So my name is Alyssa, and for my project this semester, I looked at the effects of color wavelengths on the growth and development of sunflowers. And so I'm sure everyone in here is familiar with sunflowers, but I just wanted to talk about their natural history for a quick second. So sunflowers originated in the Americas, and they were actually domesticated by Native American people who lived in the South and a little bit in Mexico. And they were actually grown as crops. And we have found seeds of the domesticated plant dating back from as early as 2100 BC. And then a cool little fact about sunflowers is that they exhibit heliotropism, which is when the young plant basically follows the direction of the sun all day long. And then as soon as the sunflowers bloom and produce a mature flower, they will stop exhibiting this heliotropism and will typically face east all day long. And so there are a lot of things controlled by wavelength in plants, for example, like phototropism and telling the seed when to germinate. But for this project, I mostly looked at the photosynthetic pigments, most notably chlorophyll A and chlorophyll B. And so chlorophyll A is the primary pigment, and we can see over in this graph that chlorophyll A primarily absorbs the red wavelengths right here. And then we also looked at chlorophyll B, which absorbs the blue and violet wavelengths. And so chlorophyll A is the primary photosynthetic pigment in plants, and then chlorophyll B is just an accessory pigment. And I couldn't find the exact ratio of chlorophyll A to B in sunflowers, but I did find that in rice plants, there's a 3.5 to 1 ratio of chlorophyll A to B. And then as we can see over here in the green section, green light is not really absorbed by either, which is why most plants actually appear green. And so with this being said, my hypothesis was that I believe plants grown under the red light will display the most rapid growth, becoming taller and displaying true leaves faster than the other treatment groups, while the control group will contain the overall healthiest plants of the study. And so to set this up, I had 7 seeds that were germinated with a paper towel in a Ziploc bag. And so 7 seeds were placed in each of the 4 groups. We had the red treatment group, blue treatment group, the green treatment group, and the control group. And then to filter out all other colors of light for each of the treatment groups, I put all the plants in a box with a colored filter on the top. So for example, if it was in the red group, it would only be getting the red light in the box. And then same for the other treatment groups. And every 4 days, I measured the height from the soil level to the apical meristem, the number of the true leaves, and the length of the first pair of true leaves. And then so here's the setup. You can see the boxes, and then I had these filters over the top to just let in a specific wavelength of light. And so for the results, we can see that all of the treatment plants actually were a lot taller than the control group. So you would think that because they're taller, the treatment group's probably healthier, right, and growing faster. But if we look at the number of true leaves on each plant, we can see that the control group is a lot higher than the treatment groups. And throughout the whole month and a half that the study was done, none of the treatment plants grew more than 2 true leaves. We can also see for the average leaf length, the control group was a lot larger than the treatment groups. And the treatment groups didn't really grow that big, as we can see in this photo. So in the very beginning, they all kind of look a little bit the same. We can start to see that so these are the 3 treatment groups right here. They're starting to get a little bit taller. than the control group. And then for the final day of the study, we can see that all the treatment groups are a lot taller than the control group, and the control group overall looks a lot healthier than the treatments. And also, I just wanted to say, the only time that the plants were not in their boxes was when I was taking the measurements and taking the photos. And other than that, they were outside all day long in full sunlight. And then here's just another overhead shot of all the plants, which makes it even more of a stark contrast between the control group, which looks really healthy, versus the treatment groups, which are not doing too great, especially the green. And then in addition, I also repeated the rate of photosynthesis experiment that we did in lab a couple weeks ago with leaf discs from this control group to measure the rate of photosynthesis for all of the treatment groups. And we can see that the control group and the red treatment had the highest rate of photosynthesis compared to the other two, which pretty much confirms what we already saw in the previous photo and what we expected, really. And so ultimately, my hypothesis was correct that the control group would be the overall healthiest group. And of course, the green light group was the worst faring group because green light is not really absorbed by plants. So the green control group pretty much died really fast. And then the control plants also grew really tall, as we can see, especially here. They grew really tall in order to try to reach the light because in the boxes they weren't getting adequate light. And this is called etiolation, and etiolation also slows the formation of chlorophyll, resulting in the yellow leaves. I think it was a little bit hard to see in the photos, but the treatment groups did get yellow leaves pretty fast. And then as time went on, the lack of energy from the sunlight stressed the control plants, and so they, pretty much didn't have enough energy to continue living, and most of them died. And then here are my sources. Thanks for listening.